Systemic lupus erythematosus, or just lupus for short, is notoriously hard to pin down. The main issue is that its symptoms and its underlying genetics are wildly heterogeneous. You can kind of imagine the immune system as a terrible rogue detective that suddenly starts attacking innocent bystanders. But the evidence it leaves behind completely changes depending on the person it's attacking. In the medical world, a disease that presents that differently from patient to patient is basically a diagnostic nightmare. And you can imagine how incredibly frustrating it is for patients who can literally go years suffering from debilitating symptoms without ever getting a definitive answer.
So here is our investigation briefing for today. We're going to cover the lupus mystery itself, why early genetic markers turned into dead ends, the breakthrough transcriptomic and approach, a highly specific 5-gene signature, and finally, what all of this means for the future of diagnosis.
Let's start right at the beginning with the lupus mystery. As you might already know, SLE is a classic systemic autoimmune disease. The immune system just goes completely haywire and triggers inflammation across multiple organs. We're talking the skin, joints, kidneys, central nervous system, you name it. Because these clinical manifestations are so varied, doctors have heavily relied on anti-nuclear antibody, or ANA tests. But there's a pretty big catch here. While a positive ANA test is essentially a required entry criterion for modern diagnosis, it's highly sensitive but often lacks specificity. Why does that matter? Well, because patients can go years without being properly diagnosed. SLE is so genetically diverse that there really hasn't been a single universally accepted diagnostic criteria that works perfectly for everyone. Scientists originally thought they could crack this by finding a specific mutated gene responsible for the disease, but they hit a massive roadblock pretty quickly.
Which brings us to the complication in our story: genetic dead ends.
Get this. A specific genetic footprint for lupus can completely change depending entirely on a patient's ethnicity. Researchers were looking closely at toll-like receptors, specifically TLR7 and TLR4, which are pattern recognition receptors in our immune system. A massive meta-analysis found that a specific variant, the TLR7 RS3853839 polymorphism, is strongly associated with SLE risk in Asian and Arab populations. But here's the kicker. That exact same genetic variant showed absolutely zero association with SLE risk in European and Latin American populations. Other variants, like TLR4 RS4986791, similarly showed no link in Europeans. This demographic bias essentially renders these static genetic markers completely useless as a universal diagnostic tool.
So looking at static DNA blueprints just wasn't cutting it. Trying to predict the disease by just adding up a bunch of different genes, which is what polygenic risk scores do, falls apart if your data only comes from one specific part of the world. For SLE, predictive ability is heavily restricted by a lack of trans-ancestral mapping. And honestly, expanding cohort sizes to the hundreds of thousands just to find these rare mutations isn't realistic for SLE. The field desperately needed a new perspective.
That desperate need for a new perspective brings us to the transcriptomic approach.
Since the static DNA blueprint was way too varied across different human populations, researchers decided to look at what the cells were actually doing in real time. Enter transcriptomic analysis. Instead of looking at the static DNA you were born with, transcriptomics looks at gene expression. Think of it as setting the dynamic mRNA work orders being sent out within a cell right in that moment. Essentially, it doesn't matter what your underlying genetic blueprint looks like. If you have active SLE, your immune system is malfunctioning right now. By zooming out to look at widespread gene expression across massive datasets, scientists realized they could potentially find a universal signature of the active disease, entirely bypassing those demographic and genetic biases.
To make sure their findings weren't just a fluke tied to a single hospital or a single demographic, researchers systematically searched existing genomic databases and combined 3 massive discovery cohorts, totaling an impressive 1,222 patients. By running a forward search, they identified differentially expressed genes between SLE patients and healthy controls. But crucially, they didn't stop there. They took whatever signature they found and rigorously tested it against 6 totally independent validation cohorts, making up another 469 patients.
All of this exhaustive detective work finally revealed what we'll call the 5-gene signature.
Out of literally thousands of possible gene expressions, a very specific 5-gene signature emerged that effectively and consistently distinguished SLE patients from healthy controls. And these genes are definitely not random. The first three, IFI27, IFI44L, and Annex A3, are strongly associated with highly expressed interferon, which is a major player in the body's inflammatory response. Then you have the ABCB1 gene, which encodes a drug efflux pump previously detected in autoimmune diseases. And finally, LY96. This is actually a newly discovered marker for SLE uncovered right in the study. It codes for a protein that binds to toll-like receptor 4, connecting the receptor to key signaling pathways. Together, these 5 dynamic work orders form a crystal-clear picture of active lupus.
And the math backs this up beautifully. In diagnostics, we use an ROC curve to measure accuracy. Basically, the steeper the curve arches up to the top left corner, the better the test is. In the discovery cohorts, this 5-gene signature hit an extremely impressive area under the curve of 0.93. So this isn't just an interesting biological theory, it is mathematically robust.
But honestly, the real victory of this 5-gene signature is its resilience. Remember that flawed TLR7 genetic marker we talked about that only worked for Asian and Arab populations? Well, unlike that, this transcriptomic signature proved generalizable across diverse ages and ethnicities. It even held up across different sample types. Whether researchers were looking at whole blood, isolated B cells, or neutrophils, the signature successfully cut right through the noise. And this matters immensely because it means a doctor doesn't need to worry about your ancestral background or even draw a highly specific type of cell to know if you're sick. The focus is entirely on the shared biology of the disease itself.
So what does this actually mean for the future of diagnosis?
When put to the absolute ultimate test in those completely independent validation cohorts, this 5-gene signature provided a real-world diagnostic accuracy rate of 88%. That is a massive leap forward. By using multi-cohort transcriptomic analysis to completely sidestep demographic hurdles, scientists have paved the way for reliable universal clinical applications. It means that in the near future, diagnosing SLE could be as simple and definitive as checking a patient's active transcriptomic score, ensuring way earlier and much more accurate treatment.
As we wrap up this explainer, it leaves us with a truly provocative thought, right? If a disease as complex, heterogeneous, and historically elusive as systemic lupus erythematosus can be pinned down by looking at the active mRNA work orders instead of a static DNA blueprint, what else is out there? How many other elusive diseases are just waiting for multi-cohort transcriptomics to finally pull off their disguises?